Hello, everyone. Um, I see everyone's getting food, and while you are eating, and take your time. I think we have a quite nice lunch selection there, and this is probably not the first time we choose this cater, and obviously our team found the cater good, so we stick with that. It's my pleasure to be here today to co-host the Lunch and Learn event with Cactus. My name is Lei Shi. I'm currently the Senior Vice President of Biointron. My job is mostly focusing on antibody discovery, R&D operation for the company, and helping our BD colleagues, marketing colleagues in the U.S. and Europe, and we have a team of colleagues here today on site. And please find time to say hi and maybe discuss your need. I already had some conversation and interesting to see how wide the audience is. And today I'm just going to give you a quick introduction of our company, followed by two distinguished speakers, and introduced by both me and Vincent of Cactus. And without further ado, I will spend just a few minutes to give you an overview of Biointron. Biointron is in the mission of accelerating antibody discovery. So how we do this. Let's talk about who we are, because not everyone here knows our company well. And we actually established almost 15 years ago. And it is around 2016 that we really into the antibody discovery area by introducing the 2-week antibody production to the research community, starting from the sequence in your hand to the purified purified products. And since then, we have grown into a company with more than 600 people. Last time I counted, it's almost 700. And we deliver more than 120 discovery projects every year, and as well as more than 150,000 antibody samples to our customers. And now the customer base has increased to almost 300 this year. 3,000 globally this year. And we are certified for both data security as well and ESG. So our customers are from all over the world, and small and large. And we have almost all 20 of the biggest pharmaceutical companies are our customers, as well as thousands of small biotechs in China and in the US and Europe. So we have a headquarters in Shanghai, and we also have a US office in New Jersey. Our Boston office has our marketing and the BD colleagues. And there's a logistics center in the Netherlands. So in terms of service, of course, we are a pure CRO company, and what we do is we focusing on the preclinical antibody discovery phase, starting from hit generation, antibody expression, optimization, bispecific construct expression, and antibody drug conjugations to our newly developed developability evaluation platforms and the cell line generation, all the way everything before before the development stage that we all cover, and this is all enabled by our high throughput antibody production platform. So how do we do this? We have, I mean, our facility right now has more than 68,000 square meters of space. We're talking about more than 500,000 square feet of space here, and besides all the hundreds of shakers and sequencing and the PCR machines, so we have a lot of automation that's developed and custom designed for our production process, and everything starts from gene synthesis to plasmid preparation to DNA dispensing, purification, as well as sample packaging and aliquoting. Most of these equipments are custom designed by ourselves and manufactured by with our collaborators in China. So we also have I mean, these are only the workhorses to make these for making the samples, but we have also an array of instruments that allow us to do all kinds of assays, from binding assays, functional assays, to developability assays. So our main discovery platform are two. One is the VHH discovery using alpaca. We have our own alpaca farm with 400 animals year round, and this farm is frequently inspected by our collaborators, our customers, especially customers from large farmers, to meet their high standard of animal welfare and compliance. The other discovery platform we call the AbJob. The AbJob platform is based on microfluidics that we developed with using several different microfluidics instruments from instrument makers, and this process can greatly reduce the the normal IgG discovery process by 2 to 3 months. So how we actually do this? We actually after the mouse or rat immunization, we isolated the plasma B cells, and then put this into a droplet-based microfluidics instrument, and then collected how the droplet is formed is that using both the oil and the aqua phase, and you can form this nanodrops of only like less than 100 microliter, less than 100 nanoliter, and then this microdrops contain a single plasma B cell that secretes a minuscule amount of antibodies, and along with the solutions that contains labeled antigens and the secondary antibodies, and if there's effective binding between the antibody and the antigen, there will be a flat reaction, and it can be detected and sorted so we can collect those positive plasma B cells, and they went through the NGS sequencing process and they get a paired heavy chain and light chain sequences, and the rest of that is going through our high throughput antibody production process, and together to try to capture the entire repertoire from a single mouse or multiple mouse, so to speak, imagine you can obtain thousands of positive cells and the sequences, and then those sequences can be analyzed and produced in a high throughput fashion with the diversity with the diversity information in hand from the very beginning. And then the rest of it, we can go through all kinds of assays. Here is just one case showing that for a 6 transmembrane targets, I mean specifically this is probably a Claudin 16, Claudin 18.2, and from this case we actually collected about 2,500 droplets and get 250 unique sequences from NGS. And through a series of characterization and purification, we eventually get 20 antibodies binding to human, and 15 of them bind to all 3 human cyno and the mouse cell lines. So this is a very efficient process, and not only we can do this, we also can work on GPCRs and soluble proteins, single pass transmembrane proteins. And the advantage of this platform is really you can sort through millions of cells in one day and get the sequence information in a week. And within a month, you can get validated binders from the entire repertoire of the animal. A big part of our process is antibody production, antibody expression. I mentioned to you that we are the first company to offer 2 weeks production of antibodies. Nowadays, we can work on all IgGs, IgAs, different formats. I mean, different species, human, mouse, rat, and rabbits, and the VHH, of course. And then not only that, nowadays we see almost half of our customer requests are for bispecifics and multispecifics. And to date, we have experiences with almost 300 different formats. I mean, it's 260 it's a year before, and nowadays the number is approaching 300 formats. Of course, people are still geared more towards the most commonly used bispecific formats, but the imagination from our scientists and the customers are limitless, and that's why you see all kinds of different formats being worked on. So this gives us a lot of data and experience to support the increasingly complicated demand for large molecule discovery and innovative approaches. So I'm going to skip this for antibody optimization, but just saying that the optimization process is fully taking advantage of our high throughput antibody production capability, and our affinity maturation is actually a non-biased approach covering all possibilities on every CDR positions, and this process has been worked extremely well and predictable for us. That the affinity maturation project in general can be finished in 2 months and up to 1,000-fold affinity improvements. So recent days, the demand for data is increasing dramatically, especially from companies working on AI and machine learning, and not necessarily needing the purified products, but data, the data for expression, the data for stability, the data for binding, the data for non-specific binding, all those data are more important than the physical purified product itself for many of these customers. And so we have developed a process, a platform that generates data, generate the data, and in an extremely efficient and high throughput way. And if you're interested in this, I will have a little bit more in detail talk at the PAX meeting next week. And these are the assays that we can cover there. So overall, I think our service covers from hit discovery to using the expression capability and eventually try to accelerate your discovery process to get the clones that you need for downstream development. This is just a recap of what we can do. And that's all. And that's all my introduction. Thank you very much.
Thank you. Okay, so for today, I wanted to talk briefly about Cactus Bio and specifically tackling challenging targets with our advanced protein platforms. So kind of before I get started, I just want to quickly highlight some of our key research areas and the platforms that we have that help to service these developments. So we offer various transmembrane proteins that utilize our VLP or nanodisk platforms. These are full-length multi-pass transmembrane proteins. We also offer various MHCs and soluble TCRs. These include class 1s, class 2s, monomers, tetramers, as well as soluble TCR expression. Additionally, we also have a variety of cell and gene therapy reagents ranging from various ELISA kits to GMP cytokines, as well as various GMP enzymes. These include gene editing enzymes, endonucleases, mRNA enzymes, and et cetera. So in addition, we offer this new kind of ultra-low endotoxin line specifically for groups that are looking to use our proteins for immunization. This is particularly useful for not only existing targets that you can go online and kind of see that we have these targets already additionally EU purified, but I also want to emphasize that if there is a target on our website or not, we do have the ability to perform additional endotoxin purification, so we can always kind of help and assist with the ability for you guys to use our proteins in your immunization schemes. In terms of quality control, we have a variety of different instruments that range from your purity and aggregation studies to binding via SPR Biocore, or Gator Prime BLI, et cetera. And then kind of going on that, I do want to say that for us, bioactivity validation is one of the most useful ways that we validate our proteins. Specifically, we like to use SPR, ELISA, FACS, as well as a couple other methods that I'll talk about a little bit later. And here's just a good example of CD19, a very popular target. And as you can see, we have good binding with ELISA, SPR, and then we even get some good population shifts with FACS. So some of our featured products, the first of which is our TCR and CD3 complex. So what's very interesting about this is that in kind of native patient samples, you have these hidden epitopes that are kind of not present because of the way that the CD3 and TCR co-receptors bind together. And so what we've done is we've developed these nanodiscs and VLPs that are basically able to express this TCR-CD3 complex in a more native environment. And one of the kind of key differentiators here is that a lot of times for antibody generation, if you don't have this native co-receptor complex, you're going to be generating antibodies that target either non-functional epitopes or non-relevant epitopes. And to kind of prove this, we tried with two different TCR-CD3 complexes. So one, we have an IG4C113 TCR. This is an affinity matured variant that binds to the HLA-0201, specifically the NY-ESO1. So this is very common in various cancers and melanomas, And additionally, we can kind of see you get really good ELISA binding as well as BLI and SPR binding. And this is with a known OKT3 anti-CD3 antibody, or monoclonal antibody. So this shows really good kind of native binding conformation. Additionally, we've also done this with a biotinylated KRAS G12D TCR targeting the KRAS epitope or neoantigen. So again, really, it's kind of proving that we can, get good validated binding ELISA, SPR, BLI, and specifically, we're getting binding with the more native CD3 epitope. So in terms of kind of additional analysis with our peptide MHCs, specifically using these TCR CD3 complexes, we actually had a other company that used our products kindly share this data. You can see kinetic binding for, on the top left, we have the NY-ESO1 TCR CD3 complex nanodisk, and then below we have the KRAS complex nanodisk. And you can see we get really good, nice specific binding compared with the controls on the right, where you see you kind of have a lack of unspecific binding in general. Similarly, we see kind of the same idea when we are using a monovalent UCHT1 in the MTAC context, or in other words, basically we have this engineered T cell receptor that is bound to a CD3 arm. So it kind of helps identify these cancerous antigens while also pulling the CD3 to kind of promote intracellular signaling. And then as you can see, on the top line, we have these different kinetic binding studies kind of at your baseline 25 degrees Celsius, but then we also see retained activity at a more biologically relevant 37 degrees Celsius. So kind of in general that I've kind of talked about some of these popular VLP and nanodisk membrane protein complexes. I do want to kind of summarize what each type of platform can be used for, specifically which ones we kind of see are validated well versus which ones we don't really recommend you use it for. Specifically, both of these transmembrane platforms are really good in your ELISAs, SPRs. Some VLPs we don't see do as well in BLI, but generally the nanodiscs do well in BLI. For FACS, again, we can kind of see some are hit or miss, so we really recommend that you kind of reach out to us and we can kind of help and see if we have any case studies that might prevent unnecessary experiments. And then similarly, these GPCRs, 4-HBs, ion channels, and solute carriers are all very difficult to express drug targets. And we see really good results with our nanodiscs as well as our VLPs. The one caveat being ion channels, we don't really recommend VLP just because the structure of that protein is very critical, and really nanodisc is kind of the way we would recommend to make sure you get that functional native transmembrane protein. So these are not only platforms that we use for catalog products, but we can also custom produce. So these are just some of our popular targets that are already on our website. You could add them to your cart and check out later today if you wanted. But we also do custom production, whether that's you providing us with a UniProt ID or an amino acid sequence, or even a literature publication that we can look into. So now I've kind of talked about some of these protein platforms. I also wanted to talk about this kind of emerging KLK target. So the KLK protein family is essentially a very popular target for a variety of different cancer targets, immunology targets, and can be a somewhat reliable biomarker for a variety of different disease states. So in terms of kind of really digging down to what the KLK is, these KLK proteins exist in both a pro form as well as an active form. Essentially, the active form is when this pro sequence is cleaved off enzymatically by thermolysin. And this typically can happen, depending on the KLK, it can either happen via self-activation, or it can be activated with the addition of a catalytic enzyme, like thermolysin. Similarly, it's interesting to note that for KLK5, for example, we can see that this KLK protein actually self-activates not only after being recombinantly expressed, but also in kind of in ourselves, in the human body. And then kind of on the converse side, we see other KLKs like 6 and 8 that can actually auto-inactivate. And then just on the right, you kind of see a general cascade overview. So then in terms of why, this direct pro form versus active form, what does that really mean in terms of kind of antibody generation and overall relevance? We find that immunized rabbits with recombinant active murine, KLK5 or 7, we actually see that we can identify these good anti-KLK antibody binding specifically with the active form. And so what's kind of interesting about this is this idea of introducing removable protection, where you can have an antibody that is targeting a specific KLK protein, but only in its active state and not in its inactivated or pro state. So in terms of how we actually go about providing both the pro and activated proteins, we have a couple different ways that we can do this. One way is that we can recombinantly express the active form and make sure that we include it in a buffer or solution that is going to prevent any type of auto-inactivation. Alternatively, we can recombinantly produce the pro version and introduce some thermolysin enzymes, for instance, and we can get that resulting active form. In general, we usually recommend the active forms for anything that has to do with antibody generation, because that is going to be a more biologically relevant state. And then in terms of kind of overall, why KLK, what does this have to do with my research? We actually see that these KLK proteins are very, very abundant in a variety of different disease states, both as biomarkers as well as kind of critical proteins for kind of managing the homeostasis of these specific cells. So for example, we can see there's a lot of overlap between certain KLKs, and they really do cover various different areas, from cancer to tumor microenvironments to inflammation and immunity, from desquamination all the way through to cardiovascular diseases. And then I also wanted to talk about domain proteins, kind of, so now that we've talked a little bit about KLKs and how we can have these active and pro forms, I also wanted to talk about domain proteins, specifically our cadherin 17. So this is a very interesting target because we see a lot of different pharma groups that are developing ADCs, T-cell engagers, bispecifics, particularly against different domains of this protein. So the CD17 is predominantly expressed in epithelial cells of the small intestine and is a key biomarker or key target for GI tract cancers. And in order to kind of help with these developments and antibody generations, we have a suite of different specific domains for that cadherin 17 protein. And I do want to say, there are other domain proteins kind of similar that we do have, expansive offerings, but I did want to focus on this just because, we can get really good data with anti-CDH17 antibodies that target different domains, and each domain will have a slightly different kind of clinical relevance. And then lastly, I just want to talk about some of our popular tumor-associated antigens. So one right now that's very popular is our STEAP1, or the 6 transmembrane epithelial cell pass, very popular for various T-cell engagers and ADCs, although in theory it really can be developed with vaccinations in mind, as well as different CAR-T therapies. And then here, just kind of again, good data validating that we do have VLPs for these proteins, and we are developing nanodiscs for both STEAP1 and STEAP2 currently. So if you're kind of interested in that, I'd be happy to connect offline and kind of update you on where we are with that. And thank you. And I'll have Dr. Lei come up and introduce his speaker.
Thank you very much. Yeah, now it's my great pleasure to introduce our next speaker, Dr. Shashi Jatiani. And I know Dr. Shashi from various encounters during conferences, and this will be probably the first time to hear your research in detail. And Dr. Shashi completed his postdoc training at the Temple, which I live in Philadelphia for many years, and he got his PhD in molecular biology from the Tata Institute at the University of Mumbai. And so he has been doing a lot of oncology research in biotech, and as well as being a faculty at the Mount Sinai Medical Center. Dr. Shashi, welcome.
Thank you. It's a pleasure to be here. I'm very grateful to the organizers, for my friends who reached out. I'm so happy this has worked out for today. Also very grateful for everyone who took the time to be here. You could have been in another place at this point, but you chose to be here, and I'm super grateful for that. I want to talk to you about comprehensive Fc-Effector function profiling of approved antibodies, the two very famous targets, CD20 and HER2. But I want to also talk across formats, because the technology platform that I represent from Seromic Systems is therapeutic area agnostic, antibody format agnostic. The only thing we need is an Fc. So it could even be an Fc fusion protein. But before I go there, I just want to tell you how coherent this progression of speeches is. You began today by listening to someone who represents a company that's making headway, not only making antibodies fast, but making them with high quality, making them for actual therapeutic development and for research that is therapeutically relevant. Then you transition to listening to another great speech from a company that prides itself in antigen and protein quality. And it's very important to understand that unless these things come together, development will be riddled with poor quality and poor metrics and unpredictability. So where do we fit in this larger landscape is when we work with both antigens and antibodies. We don't make antigens, we don't make antibodies. Developers do, or their CROs do, or their CDMOs do, but they send it to us for testing. And what do we test? We're not obsessed with, but focus on the Fc. We're probably the only company that's a service CRO in the world that does only Fc-Effector function. So why so narrow? Because it may sound narrow, but it's actually broader than just what we may think Fc function is all about. So I just wanted to give you that 30,000-foot view, and now we're going to get into the few hundred-foot view and then really down on the ground level with some cool data that we've generated over the last couple of years. I do direct strategic partnerships, but I used to be director of research, so kind of a technocrat. This is the outline of my talk, and I've asked our dear friend to wave at me at 20 minutes because I don't want to overstay my welcome. I have 30 minutes to talk to you. I'll briefly introduce the antibody Fc region in context of the Fab. We don't think they work independently of each other. There's this notion that you can develop binders and then just plug an Fc and know what you're doing with it, but we have learned otherwise, and the field has learned otherwise, and we'll talk about that. I'll talk to you a little bit about what the landscape is as far as Fc-containing antibodies, because we do talk here about Fc-independent antibodies and binders, as reagents and as therapeutic modalities. Then I'll get into Fc functions, which is what our focus is, and how complex it is, and why we're not at this plug-and-play stage where you just feed a sequence and out comes the prediction of what the Fc function will look like for the various immune cell types. We hope we get there one day. We made a start from the Botnar Institute in Switzerland, where they came up with Fc-GPT. Anybody heard of that? Right? That's a great start, but it doesn't help us yet because there are limitations and it's not enough of a data set to have a validated large language model like that. So data is king, always. Data is key. Quality data is key, and that's what we're going to tell you. Then I'll introduce our platform, not because it's a sales pitch, but so that you know what is the promise and the premise on which we stand. And I'll show you 3 case studies, approved anti-CD20 mAbs on our platform, trastuzumab, and its 2 ADCs that are approved. The first 2 that were approved, there were more approved. And then HER2 targeting, CAR-T directing, Fc-engineered mAbs. This is a fantastic new idea with a collaborator from UPenn, and I'll tell you about that. And finally, I'll summarize as to why Fc function, if done well, can be a powerful ally in therapeutic antibody development. All right, so most of you know the left side of this very, very well. I'm probably the poorest on that knowledge in this room. Is finding the binder, finding the specificity, finding the affinity, finding the developability, which is why it's such another cool progression, because Lee's going to talk to us about developability of the whole antibody, not just on the Fab side, right? And then Fc region is where we focus. They both are connected. It's a whole molecule in nature, and they do talk to each other. They are not independent, they are interdependent. The Fab influences the Fc, and the Fc influences the Fab, so we don't like to break up this beautiful marriage. We like to keep it together and talk about them as partners. And we know that the Fc region is also a target of engineering, just as the Fab region is. And the focus is on 3 different types of Fc interactions. There are receptors on immune cells. There's also soluble Fc binders, such as the complement system. There are neonatal Fc receptors, FcRn, which is mostly for PK from our developmental perspective as developers of antibodies. And then there's the Fc receptors that are on cells are mostly, the focus is gamma receptors, right? And I'll show you a good amount of data on gamma receptors. They're crucial for therapeutic efficacy and safety, these interactions and the outcomes of these interactions. And they mediate a host of physiological activities. So many times we talk about what is our antibody doing to its target? But where we stand in this whole overview is, what is the body or the immune system going to do to the immune complex when it's formed within a human body, right? And that's where this really comes into play. It can predict, or at least alert, clinical outcomes and safety, both from safety and efficacy perspectives. In development, 95% of the antibodies that are in approved or late-stage development have an Fc. So this is not irrelevant. It's not like we walked away from Fc and this is an arcane thought here. It's very relevant. Almost everything in the clinic, barring 5%, almost everything in development, barring 5%, has an Fc in it. And there are various isotypes, but of course, IgG1 is the superstar of all these things. It gets a little bit complex, and I want to point that out because it's very important to know that empirical testing will give you data that's context and disease specific and relevant for development. And why is it complex? Is because if you look at that blue sphere and consider the target cell that the antibody is trying to get to, and the red dots around it is the antigen, and the green antibody is decorated, maybe not in the same valency and density, but this is just representation. Look at all the number of effector cells and complement that come to the Fc if the Fc is not mutated in any way, shape, or form. And you know the majority of approved antibodies with an Fc have a wild type Fc. Now, of course, that I do, but can you really say I mutated 1 residue or 5 residues and have silenced it? I don't think so. Can you say that I have done this before for another target with a different epitope and we have enhanced NK cell activity? And so we can just use the same strategy for this context with a different epitope? I don't think so. And why I don't think so will become clear as the data shows up. It gets a little more complex because not only are there various effectors, each one of them have a different subset of receptors that they express. And these receptors are expressed sometimes in an inducible way. And so it's very disease-specific, very biology-specific. And one cannot predict this just by doing a few potency assays or a few binding assays or just a few guesswork assays in terms of what that stoichiometry really is and when it'll dial in. So empirical testing is king, is what we believe at this stage. I know we're going to come to a place, and that's my dream, where I'll feed a sequence into a large language model and tell me, in this disease context for this target's epitope number X23, you're going to get this much function out of neutrophils, so much out of NK, none out of complement, and you're all good to go because that's your strategy. That's our platform. In the pink, what looks slightly yellowish, depending on which angle you're looking from, the 4 rectangles are our biophysical assays. Some of them are relevant for purified therapeutic antibodies. Some of them are for serum samples, and so this platform is really good for vaccine developers also. You have antibody Fc receptor binding array. We do all the gamma receptors and FcRn at different pHs. Antibody isotyping and subclassing for polyclonal samples such as serum, because you really need to identify, and this is really good for disease biology also and for clinical trial correlates when you have patient samples coming back from trials. And then, of course, we do antibody avidity by SPR for polyclonal and affinity by SPR for purified samples. But what we do very specifically differently is we always do it in the presence of antigens. So our SPR, for example, is developed as a tripartite assay. You have the FC, you have the FC receptor, we also have the antigen present. And that's rare but very relevant in the field. In the blue are our cellular or our functional assays. Complement's not a cell, that's why I say that's the acellular functional assay. But we're looking at monocyte-dependent phagocytosis, NK cell dependence, ADCC, complement activities by two different assays, dendritic cells, which you know sit as a bridger between innate and adaptive immunity. We are also looking at neutrophils, which are the most abundant cells in the bloodstream, and we somehow forget about them when we do Fc-Effector function, right? We look at eosinophils, minor population, but powerful cytokine secretors can cause issues with toxicity. Basophils similarly, and then we have another assay for NK cell activation. And the last two, mucin binding and enhancement of infection, is really more relevant in the infectious disease context. Our platform is therefore physiologically relevant in the presence of antigen. We use human cells only, CP assays with everything, and then we have also primary cells. We actually get cells from healthy human volunteers, and we've made assays that are so robust that our CVs are quite acceptable and go directly into regulatory filings because we are a GCLP accredited facility right here in Woburn, just outside Boston. Right? We are high throughput, can do a few thousand samples in a single experimental run. These are all plate-based, mostly bead-based assays that are very acceptable to FDA. We've had conversations and webinars that we presented at the FDA. We also have a computational pipeline, because if a client comes, they don't have to. They can do just 1 or 2 or 3 of the assays or 4 of the assays. But if a client comes and says, let's do the whole thing, because they really want to understand that drug product, that's a huge data set. So we have a computational pipeline that will help you deconvolute and not guess what it means, but have statistics tell you what it really means. First case study, CD20 mAbs. 3 approved antibodies. Of course, there were many more to follow on. Rituximab, ofatumumab, obinutuzumab. We know very much about them since 1997, or if you were following the preclinical data before that, or were alive back then, or as a reader of science back then, you know about these antibodies. They have different epitopes that they bind, depicted in those wonderful different colors. Ofatumumab is the strongest binder because it attaches itself to both extracellular domains 1 and 2, and therefore predicted to be the strongest effector function inducer. The test articles in this study are going to be of the 3 antibodies, and controls are a reduced function NQ version where the glycosylation is taken away, an isotype control A2, which becomes very important in one context, and an anti-Ebola irrelevant antigen mAb as a negative control. Here is biophysical binding followed by functional activity. There are 2 alleles of FC3A, Fc gamma receptor 3A. It's V168 and F168. One's a higher affinity, the other is a low affinity. These antibodies bind, and the controls look good. They bind at different levels, and this is actually in line with canonical information in literature about these 3 antibodies. But look how predictable is the rank order for NK cell-mediated Raji cell killing ADCC assays on our platform. Complete match, and I think it's so good is because we have a good understanding in the presence of the antigen in the biophysical binding. Similar outcomes in terms of predictability of biophysical binding for complement-dependent activity. C1q binding on the left as a rank order, ofatumumab is the strongest inducer amongst these for complement activation. You have antigen-specific ADCD, which is deposition of complement in the absence of effector cells in the same rank order, and lo and behold, Raji cells being killed in the same rank order in a CDC assay. 2A binding is very important for ADCP activity. Again, there are two alleles here, one high affinity, one low affinity, H131 and R131. And you see binding at a fairly robust level for all three antibodies and activity at a fairly robust level for all three. These three antibodies actually induce roughly the same level of phagocytic activity. But notice the green bars. Suddenly, the NQ is binding one out of the two alleles. So I went back into literature and said, why is that happening? Guess what? The cells we are using here is THP1 cells, and they are homozygous for the R allele. Picks it up, shows up in the activity. 3B is, receptor 3B is very important for neutrophil, as is, folate receptor alpha and neutrophil have these being expressed on them. So we looked at both of these for neutrophil activity. As you can see, robust binding in a slight rank order for 3B. Receptor alpha, of course, will bind only the IgA variety of the isotype. This is where it becomes relevant. But look at the look at the functional outcome in neutrophil-mediated phagocytosis. The rank order is again maintained, but you see the highest activity through an A2 isotype. And these assays tell you that you can predict certain things. The outcomes tell you that this is completely understandable. What's really cool is not all of this was known. Nobody had tested the bottom half of that graphic. Nobody really had tested neutrophil phagocytosis for these, and now we have. And these ancient, established therapeutic agents are showing activity that we did not know about because we have a platform that can assess it. And also eosinophil phagocytosis. Eosinophils are difficult cells, like I told you, and there's some lung injury related with at least rituximab, where you can trace it back to eosinophilic activation. And there we have it. If people had done this earlier, maybe they would have chosen a rituximab 2.0 and not rituximab. This is a picture I love. You know, there's a company that came up with the periodic table of antibodies. I love them for doing that. Everything in a blue rectangle here has an FC. The things without an FC are antibody fragments. But there's been an explosion of formats. Don't we know that? It's not because people are having fun just for the sake of having fun. Some diseases are difficult to treat. Some targets are hard to reach. And you need to switch the format because you don't know what will be the best in your therapeutic outcomes. And so you do this. There's conjugates, there's engineered, there's polyspecifics, and there's isotypes, and then there are hybrids of these formats also. I don't want to spend a lot of time. I'd spend the rest of this afternoon just talking about the different formats that we're dealing with. But because our platform is format agnostic, we can test all of them. I'm not going to bore you with the literature survey that's on this table for trastuzumab and its ADCs. But overall, this is the plan. Test trastuzumab and its ADCs, and the controls are very relevant. IgG1 negative control for trastuzumab. IgG1 TDM1 with the same DAR as Kadcycla as a control for TDM1, and TDXT similar story, and keeping, again, a negative control antibody that should not bind the antigen. 2AR, 2AH binding looks great. 2B binding, which is the only inhibitory receptor, also looks great, even though it's less. These are not exactly comparable. Please don't do that to yourself. Just know that the ranking is important. The 3AF, which is more for NK cell ADCC, is bound in a certain rank order. And it tells you that TDXT should be the worst in eliciting NK cell mediated ADCC. And that, by the way, is known in literature amongst these three candidates. And then 3B, which is, like I mentioned earlier, for neutrophil phagocytosis, there's a rank order of binding. All very well, FcRn binding. There's a rank order at pH 6, and that matches the known half-lives of these antibodies. Trastuzumab lasts the longest, TDXD lasts the least, and TDM1 somewhere in between. And there's no binding at pH 7.4, which means recycling is not affected. You can go on with predictable outcomes here. The engineering of adding the drug payload has not messed up recycling. And then receptor 1A is prerequisite for all Fc-Effector function, even though it does not really differentiate between which cells get triggered or not. It is the high affinity binder, which is always present when Fc function is elicited, and we see that they're roughly equal in that. This is the type 1 high affinity receptor. We got a huge surprise when we looked at C1q binding. Look what happened to the blue bar in that data set. Trastuzumab, not known to elicit much complement activity, and TDXC even less than that for C1q binding, but TDM1 goes through the roof, and that kind of threw us on a head spin because that's not known in literature. We went in, I did a literature survey, and we found out that because of the payloads and because of the chemistry of the payloads and the biophysical nature of those chemistries, you have different things happening in terms of hydrophobicity and the kind of docking that takes place of the Fc receptor on these Fcs, which are now modified with drug payload. And trastuzumab is a clean docking situation. There's nothing there as a compound to interfere. TDM1 has something called TDM1 patches, DM1 patches, and that leads to a potential sticky interaction. Maybe that's what's happening here. And there's physical sheathing of CH2 in the case of TDXD, and that's why you've seen reduced binding here. So what's the tiebreaker? Well, let's go back to the ADCD assay. We do that, and boom. The green bar begins to behave like the red bar. And so it's important to understand that chemistries also make a difference in biophysical assays. It's not always predictable if you don't understand the chemistry. We'll jump now to the last part of my case studies. I don't know at what minute I am, but I think I'm doing okay. Jason Lohmuller's lab in UPenn has this really cool technology. Look at that antibody, which is an anti-HER2 IgG mAb, and he looked at both trastuzumab and pertuzumab as starting points, added that black linker to it, and then attached a synthetic motif. What is a synthetic motif? The synthetic motif is what actually is recognized by a CAR T cell that's engineered to recognize that synthetic motif. So what we were doing is trying to de-risk or predict what will happen when you come through this very complicated 4-molecule 3-cell interaction in this strategy. These are antibodies modified to direct CAR T cells to the target cells expressing HER2. You have immune effector cells with the blue Fc receptor. You have CAR T cell with the purple CAR T that's specific for the motif. And you have the red cancer antigen on the cancer-expressing cell. And what's bridging them is this cool engineered antibody. And we are trying to look at the difference between various lens flexibilities and sites of conjugation. So we put that on our platform, and I'm showing you a subset of things because it gets really busy really fast and gets very difficult to see. So just 2 modifications, one representing random lysine conjugation in red, the other one site-specific conjugation. And what you see is in blue are the parental mAbs, trastuzumab on the top panel, pertuzumab on the bottom panel. They have activities that are expected for 2A binding, 3A binding, FcRn binding at pH 6. 7 looks good. I'm not showing it to you. But modification 2 is the one that fares better in terms of binding these receptors and therefore predicts better for not causing unnecessary immune activation or CAR T cell fratricide. The immune cells come in and kill the CAR T cell. You don't want that to happen. You want all the activity in this strategy to come from the CAR T cell, not from other immune cells that can complicate your PD and complicate your safety. And so it's predicted that modification 2, which is site-specific, avoids Fc-mediated immune toxicity and prevents CAR T cell fratricide. So did that happen? Let's go to the next part of it, which is the Fc-Effector function. I'm showing you several functions here. ADCP, undifferentiated monocytes, ADCC, NK cells, ADDCP, which is monocyte-derived dendritic cells, ADNP, neutrophils, eosinophils, basophils, and finally complement. And as predicted from the biophysical binding, what do you find? You find that modification 1 does poorly compared to modification 2. Modification 2 to the site-specific one is the way to go. And currently, we are in the next phase of this study where we brought the CAR T cell into this mix after doing it in the absence of CAR T cells. And we're so excited to see where that will go. I'm sure it's going to be a cool publication. Jason and his team are wonderful at UPenn. So to summarize, what have we learned from this broad profiling and why is it necessary? Well, it's not necessary because many people can do without it. You know, there are these antibodies that were actually approved and made billions of dollars at peak sales without doing the Seromics platform. We get that. But if you really want best in class, here's an opportunity, right? Why assess it early and why assess it comprehensively? Because FC biology right now is complicated, hard to predict, must be assessed empirically via a robust, physiologically relevant antigen-specific platform. We insist on the presence of the antigen, especially for biophysical binding. And if you do that, look what all you get. Deep mechanistic insights for differentiating your antibody versus other antibody competitors. Doesn't matter which format. Ensure the efficacy and safety of simple, complex formats and novel modalities, such as the one I showed you right at the end. Validate the impact of Fc silencing and enhancing mutations, because there are assumptions that you make a certain set of mutations and you're going to enhance it or reduce it. There's a whole other data set I'm not presenting today where we put a slight question mark, not a cloud, a slight question mark whether there is something like a plug-and-play Fc or not. I think there is not, because the epitope's distance from the membrane of the immune cell, how proximal it is, will determine how strong a function you elicit from that immune cell. Also, the antigen density will cause, if it's higher density, more Fc tails to cluster. And when the clustering of the Fc tails happen, you actually trigger function. So every antigen, and therefore its antibody complex, is unique in its own right and must be studied empirically. And there is no plug-and-play Fc mutational strategy that I think I can put my hat on, and I'll take it on me. I know many others who take that, and they do well. Validate the impact and enable data-driven, clinically aligned decision-making for candidate selection. You're selecting a candidate. You're going to go into NHP talks. That's a huge jump if you don't know what the efficacy, minor issue at that point, or the safety looks like. De-risk the safety for NHP talks, and you have a strong path to the clinic. And that's what this is at. And once you enter the clinic, if you already know mechanistically what to expect, you'll be well placed to have all the correlates in hand for not just efficacy, but also the immune correlates that you can predict and assess. And finally, we support regulatory submissions across developmental stages. We have been at IND stages with our clients. We've been at also BLA stages and the things in between. Well, that's my talk. Just wanted to tell you a little bit more. We are actually a thinking partner of our clients, because this is just since 2025. A review in mAbs, a paper in Nature Communications, collaboration with King's College London, another paper with the same group in cancer research, and guess what? We just got accepted in Cell. It's going to come out at the end of this month on the 28th. So we're not the typical CRO. We are deeply thoughtful about the science that we represent, and we are thinking partners of our clients. So history, I'm going to skip. We were founded in 2019. Galit Alter is a co-founder. She was right here at Raygon, Harvard, MIT, Mass General. And this came out of that. Those are the papers that gave us a right to become a company. She's very well published, very well known, mostly in vaccinology with polyclonal samples. We have taken that same platform of the set of assays, made a robust industrialized platform out of it, and now becoming very applicable for therapeutic antibody development. Those are the people who work for us, an excellent team. I truly love working with each one of them. Abigail's right here with us at the back. And these are our partners who let us disclose them as our partners. There are many more. Thank you.
All right. Up next, we have Lee Andrews, who began his career as a raw materials specialist at Johnson & Johnson in New Jersey before transitioning into a bioanalysis manager role at Syneos Health, formerly Inventive Health Clinical. During his more than 7 years at Syneos, Lee led the bioanalytical oversight of numerous projects, managing both internal operations and external client relationships while driving successful project execution. He currently serves as Associate Director of Bioanalytical Development at Vistera, where he has spent nearly 6 years advancing bioanalytical strategy and development initiatives.
Thank you, Jordan. Yeah. And I want to thank the sponsors for organizing this event and for inviting me to speak on a topic of interest to me and hopefully of relevance to all of you as well. First up, my disclaimer slide. I work for Vistera, as was mentioned. They're a fully owned subsidiary of Otsuka. Oh, it is auto-advancing. Hold on. There we go. The methods and data presented are the intellectual property of Vistera, but the opinions and conclusions expressed are my own and may not be shared by the company as a whole. All right, with that done, here's a quick look at the outline for today's talk. I'm going to be going through immunogenicity and the critical decisions in the design and development of neutralizing antibody assays. Hopefully that's relevant to this audience as well. First of all, I'll be giving an introduction to immunogenicity and to neutralizing antibodies, as well as to the Vistera compound VIS-171. We'll then move on to an overview of the assay format that's in use for the clinical trials, as well as a couple of the key decisions that went into the design of that assay, namely selection of the agonist for the cell line and selection of the surrogate positive control for monitoring the assay's performance. We'll wrap up with the customary observations and conclusions. So first up, what is immunogenicity? Well, immunogenicity is very complicated, as anyone who studies it knows, but we're going to look at a very simplistic kind of definition for the purpose of this talk. That being, it refers to the ability of the immune system to trigger a response to a substance which the body views as foreign. We're going to further simplify it by basically putting that into two different categories. First, the activation of innate immune cells, or the adaptive production of antibodies. For this talk, I'll be focused primarily on the latter half, specifically looking at neutralizing antibodies and how we measure that response. So why does it matter? There's many reasons why immunogenicity matters, but primarily it comes down to safety and efficacy. You can have a myriad of unwanted immune reactions to your therapy, including allergic responses such as anaphylaxis. You can have reduced therapeutic duration if antibodies clear the drug ahead of time. You can have diminished therapeutic effect if you have neutralizing antibodies, which again will be the primary topic here. And then you can also have autoimmunity, resulting in various downstream effects, including tissue damage if you induce that. So because of these safety impacts, the presence of neutralizing antibodies, specifically cross-reactive ones, is of importance in the biologics development space. Next, we'll have an introduction to the compound, VIS-171. As you can see, it has a human IgG Fc that's been immunosilent, or at least claims to be immunosilent. Modified IL-2 moieties, or domains that are tethered onto it, providing an antibody-like fusion protein design. This is used to agonize T cells. Specifically, the IL-2 domains were modified so that they focus primarily on CD25, the IL-2 receptor alpha unit, which causes the expansion of Tregs. It should be noted that because the compound includes these IL-2 domains, despite the fact that they are modified, they're analogs of endogenous proteins, the IL-2, and therefore these cross-reactive neutralizing antibodies are the chief safety concern when thinking about using this for chronic dosing. To address that, we obviously needed to have a neutralizing antibody assay. Here's an overview of the assay format, but briefly, it consists of an IL-2 active reporter cell line. Once activated by IL-2, or in the case of the diagram here, the IL-2 domains present on VIS-171, it causes the release of alkaline phosphatase. So in the absence of neutralizing antibodies, the drug activates the cells, and you get a signal. When neutralizing antibodies are present, they cause a blockade of that, and there's no signal. This brings up the first question when developing the assay, or at least the first question that I'll be covering in today's talk. That being, what is the more appropriate agonist to use for the assay format? Now, we basically have two options, right? We have the drug itself, VIS-171, or we have recombinant human IL-2. Now, upon consulting with some different contractors to get some feedback on what the regulators expect, there was actually different opinions on this, right? Because the greater safety concern is around neutralization of endogenous IL-2 and the induction of these autoantibodies. However, VIS-171 is the compound being dosed, and so therefore it would be presumed to be the immunogen. It's also slightly modified and has a more complex structure. So we had to dig into it to figure out which one we thought was going to be a better option for the assay. To start off with, we evaluated their ability to agonize the cell line, and as you can see, both of them worked. We also included two kind of different flavors of recombinant human IL-2, one produced in E. coli and one in HEK cells, just to see if the expression system had an impact on how they were able to agonize the cells. They all worked relatively well. Slight differences, again, likely due to the modified affinity of the VIS-171, as well as the difference in structure, but all of them would have been viable options. So we didn't have enough data here to make our decision. So what do? We collected some more data. We decided to bring both the IL-2 and the VIS-171 into the next step and look at selection of the surrogate positive control. Here we tested or displayed our three of the surrogate positive controls that we evaluated for this assay, consisting of two monoclonal anti-human IL-2s and one polyclonal anti-human IL-2. They're displayed in the graph here. As I mentioned, we included both the VIS-171 agonism and the recombinant human IL-2 agonism for a comparison. Now, this is where it gets very interesting. You'll note that when using VIS-171 as the agonist, we were able to see that all three of these surrogate positive controls showed neutralizing activity of various amounts. However, when we used the recombinant human IL-2, only two of those showed any neutralizing activity. Now, this is odd because, again, each of these shown antibodies were raised against human IL-2. And yet we're only seeing one of them, or two of them, showing up in the NAb assay when recombinant human IL-2 is being used as the agonist. Now, this may be due to the fact that the affinity has changed. It may be due to the fact that this particular cell line is the trimeric receptor. And so it becomes important when you're looking at this to say, okay, well, if you were to select human IL-2, you can reasonably expect that you'll be missing some positivity in actually true positive neutralizing antibody samples, which is not what you want. Now, on the positive side, it could give you a lower immunogenicity rate for your clinical stats, but the clinicians want to know this, right? This is potentially relevant NAb to see. So with that, we made the decision VIS-171 is the more appropriate to use. In terms of the second question, we also said, okay, well, of the two remaining ones, they were both pretty well behaved. You can see that whichever agonist was used, we have the dotted line showing where they intercept for sensitivity. Both showed acceptable sensitivity. The monoclonal mAb 2021 had a sensitivity of about 10 nanograms per mil. The polyclonal AHP 1032 wasn't too far off. It was about 300. And regardless of whether we used the VIS-171 or the recombinant IL-2, you can see that they had about the same sensitivity. So there wasn't a big impact on the assay either way you went with that. So we chose to go with the monoclonal primarily just because it's available, it's a known sequence, and so it gives you that kind of longevity of your clinical program. If it runs out, you can express more. With the polyclonal, you can be in trouble if you have to bridge to a new lot. All right, so some of the observations that we walked through. First of all, the recombinant human IL-2 was a suitable agonist for the cell line and did produce a robust response. There were commercially available polyclonal and monoclonal antibodies, which were identified and shown. They did have neutralizing activity against both of them. However, some of the anti-human IL-2 antibodies weren't strong enough to overcome the affinity between the recombinant human IL-2 and the receptor present on the cells, and subsequently weren't able to be detected. The antibodies, which can neutralize both, showed similar sensitivities as shown, regardless of the choice of agonist. And so we weren't really losing anything by choosing Vis171 as the agonist for the cell line. Subsequently, the decisions, as I mentioned, were to use Vis171 as the agonist for the cell line and to use the monoclonal positive control for the NAb to monitor assay performance. And finally, I'll move on to some conclusions. Firstly, that bioanalytical scientists have to consider the mechanism of action for the therapeutic. In this case, we were looking at a therapy that activates T cells, and so we wanted something that mimicked that activity as much as possible. Next, the safety implications of the immune response. We had to identify the risk profile and what was going to be of greatest concern, and make sure that the NAb assay we had was capable of detecting those antibodies. And lastly, the practical impact of the reagent selection when designing the assay. And that includes things like reagent availability, reproducibility, etc. Next was we have to be sure to develop NAb assays that can detect clinically meaningful antibody responses, right? Everybody wants to have a nice low immunogenicity rate. On the other side, you can develop super sensitive assays that pick up everything. But really what we need to know is which ones are going to be clinically meaningful. We don't want to miss those because that informs the decisions that the clinicians are making when it comes to dosing of patients and ultimately to the patient's safety. With that, thank you for your attention, and I'll take any questions. Okay, thanks again.
All right, thank you everyone. If you haven't had a chance to grab food, I would just recommend you go do that quickly, and then we will be moving on to our raffles. Thank you.